So the first virus we need to discuss is influenza virus, and before pandemic of that virus, known as coronavirus COVID-19, influenza virus was the most popular. It was the major problem, and a lot of discussions, a lot of talking in infectious world was due to that virus, were related to that virus. And nowadays it is crossed out by coronavirus, but still it is one of the most important and the most prevalent viruses in humans. If you talk about classification of virus, it is important to mention structure of genetic material, and influenza virus have negative sense single-stranded RNA virus, means the genome is presented by RNA. Actually, virus have several species or several types of virus. So again, it can be confused if you take different literature in different time periods. Sometimes influenza is described as one species but contain three types: type A, type B, type C. And nowadays, fourth type is identified, and recently it was reclassified into four different genus: alpha, beta, gamma, and delta. And actually, we have four different species of virus: influenza A virus, influenza B, C, and D. So it's more correct now to name different viruses as separate species, not types. So first description of influenza is dated by 1743, when serious epidemic serious epidemic of influenza occurred in Italy, and actually people could not explain why it is happening, so why a lot of people and massive spread of some disease appeared, and they thought that it is influence of some heavenly bodies or other unknown forces, and that word influence came into language and became normal. And actually respiratory diseases, not only that one but multiple respiratory diseases, can be called as influenza because it's some influence of unknown power on human. So that term became so popular. Actually, virus was named influenza, and that name is still used nowadays. Classification into species is important because they have different biological properties, and actually main number of infections caused in human community are related to influenza A. It can affect not only humans, it can cause similar infections in birds, in mammals, in different type of animals. And actually all serious pandemics of influenza were caused by influenza A virus. Influenza B is second virus in humans. It is described as well in seals. Influenza C can infect dogs and some other types of animals besides humans, but that virus is rare, and only accidental cases of disease are caused by type C. And that last type of virus, last species of virus, influenza D virus, is described only in animals, and in humans no diseases were confirmed yet. So a lot of people asking why that virus became so dangerous and so popular in the last decade. Is it something new, and was it known before? Why it became so dangerous just now? So the first thing, that virus initially was described even some 3 decades ago, and first avian flu means that virus was confirmed in birds and in humans as well, was demonstrated in 1901, means that more than a century ago. Of course, it's not something new. It is old virus with very old history, and swine influenza was registered in 1931. Again, virus from pigs came to human and caused disease. So it's not something absolutely new. It is old virus, and it was registered recently. All diseases are described and known. It's not some emergency. But of course, previous decades it was not so prevalent, and it was not so deadly to human. We had some cases, but they were mild and not so massive. And nowadays, virus became more aggressive and more lethal for humans. If you talk about why viruses have higher prevalence in winter time period, because natural properties of virus uh, so that virus prefer low temperature. It remains viable about 0 degrees Celsius for a week, maybe a little more. And if you talk about some low temperatures, and usually laboratories using freezers in minus 70 degrees Celsius, virus remains viable for multiple years. And of course, uh, cold time periods are preferred by virus, and actually heating up to 50 degrees Celsius can kill virus in half an hour. And of course, countries with very hot tropical climate are less affected by influenza pandemic because virus prefers cold temperature. So in some studies, it was shown that iodine have some effect on influenza virus. It still have no clinical significance for medical practitioners, but that property is identified, and maybe it is some point for future research or for some drug designing. So if you talk about structure of the virus, it is very unique, and virus is one of the most studied, one of the most known. So typically viruses have a spherical or ovoid appearance. In some samples it can be filamentous and enlarged significantly, but typical shape of the virus is spherical shape. Viruses have common structure. Genetic material, as we said, presented by RNA, and usually it is combined with proteins, which is described as a ribonucleoprotein or RNP. The combination is acting as internal antigen, and unique immune response can be identified to that structure. So RNA is broken into 8 pieces, and in virus, as you can see, it is present as some linear filaments. Those segments are uh, injected by the virus in the host cell, and the first step of host cell infection in different parts of RNA will be combined together in one RNA strand. So for that purpose, virus needs RNA-dependent RNA polymerase, means that initial RNA should be combined together in one line, and on the basis of RNA, again, RNA will be synthesized. As far as in host cell, in animal cells, that enzyme, is absent, because usually we have DNA as a template for RNA. That enzyme is always packed into virion of influenza. It's not shown on that picture, but always virus contain that unique enzyme, and it is essential for initiation of infectious process in the host cell. Virus again have classic structure, and spikes of 2 types are present. First one is hemagglutinin. It is prevalent in amount, have a triangular shape, and it is classified into 2 types, HA1 and HA2. And that peplomere, as it obvious from the name, have ability to agglutinate red blood cells. And actually, red blood cells are not so important for virus because it is respiratory virus. The same properties of hemagglutinin are observed with respiratory epithelium. And actually, that enzyme is essential for infection of the respiratory cell. And of course, in the life cycle of virus, hemagglutinin is required to stick to the epithelium. But in lab diagnosis, in identification of virus, blood cells can be used, and the same receptor is acting as hemagglutinin. So in classic description of virus, 15 subtypes of hemagglutinin are available, but in some last papers and some last research, one more additional subtype is described, and actually somewhere you can measure it is 16 subtypes of influenza viral hemagglutinin. It is not an error, it is a correct. So second type of peplomere, it is a neuraminidase that have appearance as a mushroom, and it's less prevalent in amount, but it is a enzyme which is required for destruction of host cells. 9 subtypes of neuraminidase are described, and both hemagglutinin and neuraminidase are acting as external antigens, and of course, antibody response is unique for different for every subtype of every receptor. Influenza virus is enveloped, and that envelope is made of 2 layers. First one is a lipid layer, which is an external layer, and actually it is the part of the host cell membrane. Virus leaving the host cell by budding, and during every budding, every viral particle will take some piece of cell membrane and use it as a coverage. So that lipid layer originates from the host cell. It's not coded by virus, it's not contain viral particles, and of course it is a human cell. It's very difficult for immunity to recognize viral particle because it is hidden by human cell membrane. Internal layer or protein layer or M layer, it is a matrix layer composed of 2 types of proteins, M1 and M2. As well, it is internal antigen, and M antibodies can be used in diagnosis of the disease. According to combination or presence of hemagglutinin and neuraminidase types in virus, all influenza viruses of type A or influenza virus A species, it is classified into serotypes. So routinely it is written as H number, N number, means type of hemagglutinin, type of neuraminidase. And if you will see anywhere that one of the most famous serotypes, H1N1, it means that virus have first subtype of hemagglutinin and first subtype of neuraminidase. So as far as H1N1 was initially discovered, described, and studied, then structure of hemagglutinin and neuraminidase were numbered as 1. So all previous descriptions of both peplomeres are numbered according to order. And actually nowadays, of course, H1N1 is one of the most famous viruses, and that serotype of virus caused Spanish flu in 1918. The same serotype, but slightly mutated, caused swine flu in 2009. And second serotype of virus is H3N2. That virus was responsible for Hong Kong flu in 1968, and actually it caused some epidemics recently. And we have not so many serotypes of virus circulating in humans as it possible. If you will count and you will multiply 15 by 9, you will understand that more than 100 serotypes is possible. But actually only few are circulating in community of humans and causing epidemics. If you talk about influenza virus B, it's not classified according to serotypes, it is classified into lines. And most important lines circulating nowadays is Yamagata and Victoria. They are marked as a B / name of the line. Usually line Victoria is included in vaccines, and that line is identified in human as the most prevalent. So some people asking why influenza virus causes so serious pandemics and why they occur so frequently. Actually, in Russia we have 2 epidemics every year. It means that twice a year influenza virus causes an epidemic in communities. And people asking what is the problem? Don't we have some good immunity against that virus? Or why it's not possible to prevent infections by one vaccine if it is the same virus causing epidemics? And the main answer is influenza virus undergoes 2 types of genetic modifications. It is changing gene regularly and sometimes even continually. So first type of that modification is antigenic drift. It is very small minor change in structure of genes which are happening continually, usually during viral replication. It is some error in transcription of viral genome because viruses don't have any mechanism of replication control. And when genetic material is replicated, some errors are possible, mechanisms are not very precise. And usually the change is so small, it will not lead to significant change in structure of the virus. And viruses with antigenic drift are very closely related one to another, and usually they have the same antigenic properties. Means that if virus caused disease in one human, then immune response caused by first attack will have very good efficacy against second type of viruses, mutated one, and disease is not really possible for the second time. Another change of genetic structure is antigenic shift. It means that change will be so significant, so major, that protein present in hemagglutinin on neuraminidase will be changed, and actually new subtype of neuraminidase or hemagglutinin will appear. And now it means that actually previous episode of the disease will induce immune response which will not cover new virus. Structure of antigen is changed, and antibody which was produced on previous type of the same peplomere will not be effective. And virus can cause second episode of the disease in the same human, even in short time period, and human will be susceptible to the disease. And that's why virus causing epidemics annually, even twice a year, because previous immunity will not work. And it is the problem with designing of vaccines, because actually you can include one serotype of the virus in the vaccine, but that virus will mutate and serotype is changed. But of course, mutation is always unpredictable. We cannot know which serotype will arrive after mutation. And we cannot design a vaccine which will be effective next year. We have some WHO which trying to predict which serotype will cause epidemic next year. They have some models, they have specific system of calculation of possibility, and they're trying to predict which serotype will cause the disease. But it's not really possible, and that system is not precise. And it is a problem because we cannot prevent disease by vaccination. We cannot know which serotype will cause epidemic next year. And that's why virus stable in the property to cause epidemic every year. Second problem, sometimes that antigenic shift will not change to appearance of new serotype. Serotype remains the same. But we said already that receptors can be specific to animal host, and some viruses are circulated only in animals or in birds and not affecting humans. The same with influenza virus. We have some viruses of influenza which are circulating in birds, and they're not dangerous to humans because we have another structure of receptor. But antigenic shift can lead to change in host specificity. And in that case, serotype remains the same, but host specificity is changed. And that thing was observed in 2009 when H1N1 virus, which initially had animal specificity, caused pandemic in human community. Serotype remains the same, but virus became dangerous to another animal species. And in that case, animals, birds, and almost any alive being on our planet can act as a serious reservoir for human diseases. And we do have that virus around us, but it's not dangerous for us. And we're living with that in common life. But then someday, host specificity will be changed, and that virus, which is already around us, will become dangerous for humans, and we will have new episode of pandemic. So both antigenic shift and antigenic drift are highest in type A virus or in A species of the virus, and that's why actually pandemics of flu are caused by type A. It is less frequent in type B and was not demonstrated in type C. Type D is recent virus and it's not actually studied. So of course, both antigenic shift and drift applied only for neuraminidase and hemagglutinin. For M protein, it's not described, and M protein is always stable. And again, antigenic drift, it is constant change which is always identified in virus, but it's not important for us. An antigenic shift for our lucky is happening occasionally, and the change is unpredictable, but usually it is observed once in a year, rarely twice in a year. Cultivation of virus, of course, is possible, and influenza virus is easily growing in chick embryo, and actually type C or influenza C can be cultivated only chick embryo. If you be asked how it is possible to cultivate influenza type A, because it is one of the most important viruses, or type B. So usually in specialized laboratories, we have kidney cell cultures of human or different animals, and viruses easily growing in specialized laboratories. But again, in routine laboratories, cultivation is not used, and usually diagnosis is done by molecular methods. So hemagglutinin have unique ability to clump red blood cells, and again, that property is common for different types of animals, and influenza virus can agglutinate more than 20 types of animals' red blood cells. And it is good for us because in diagnosis of disease, we don't need human red blood cells. We can use animal one, and animal cells will be agglutinated by human viruses as well. So it is easier and cheaper in diagnosis of the diseases. If laboratory is not equipped with specific devices to support cell lines, then common animals can be used, such as rodents, mice, rats, and actually infection can be done by use of sprays or aerosols by inhalation. And virus will accumulate in lungs in high amount, and lung tissues can be used to obtain viruses in significant amounts. But again, usually it is done only for some research purposes or for studies, but not for routine diagnosis. If you talk about clinical course, then that disease is absolutely perfect answered by any student in any exam, because all classic symptoms of respiratory infections are applied to that disease. So again, we had some problem, and it's absolutely awful thing when avian flu, and people say, "Oh, it is a bird who is the source of infection." And if it is avian flu, then bird is dangerous. Transmission from bird to human is possible, and everyone start killing birds. Or if it is some swine flu, then a lot of people trying to kill pigs because they think that it is a source of infection. So in human community, infection is transmitted from infected person by air droplets from one subject to another subject. Initially, virus was animal pathogen, but while it is animal, it's not dangerous for human. If virus will mutate and change host specificity, virus became dangerous for humans. It will circulate in human community. Animal is not important anymore. Virus already mutated, and it's too late to do something with animals. So don't kill those beings, because actually now it is human to blame. And in human community, in human population, infection is transmitted from infected person. We don't have carriage in the case of influenza. It is always man in acute phase of the disease. So influenza virus is one of the most contagious, and actually 3 virions delivered in aerosol state is enough to cause a disease. Only 3. And again, it is a problem because common practice to prevent airborne disease is wearing the mask. And very often when students sitting in a laboratory, in a classroom in mask, it is a good measure of protection. But very often they need to talk, or they just feel not very comfortable. They will remove the mask, sit for a few minutes, and then put it back. That time period is absolutely enough to infect the whole room, because with every breath you can exhale hundreds of particles. and actually 3 is enough to cause a disease. So if you're wearing the mask, you should put it completely always, and it should hide and close nose and oral cavity as well. That trend to wear the mask here when nose is open. Then mask is not a protection because all viruses will be easily delivered with air from nasal cavity, and again you will infect everyone, or you can inhale that virus with the nose, and it will be pretty enough to cause a disease. So influenza virus is almost always equal to 3. We have 3 particles delivered in aerosol. Incubation period is about 3 days, and disease occurred in 3 stages, 3 days each stage. So of course, virus will enter the host by ciliary epithelium of respiratory tract. Initially, it will damage epithelium here, and that destruction of epithelium is a good entry gate for bacterial infection. And very often, influenza virus is complicated or have co-infection with bacterial diseases. And at that point, diagnosis is more difficult because people will think about bacterial pneumonia or bacterial infection. And bacteria will hide virus and make it's more easier for virus to damage further cells. So presentation of clinical presentation of influenza can vary from even asymptomatic duration of disease to very severe and lethal pneumonia or fatal pneumonia, especially when viral infection is co-infected with bacterial one. So again, disease mainly occurs in 3 stages. Initially, it will start with non-specific symptoms such as very high fever, severe headache, muscle pain, joint pain, and in high toxemia, it will lead to prostration and some problems with mental abilities. So usually the stages last for 3 days. If you talk about some peculiarities of type B infection, then especially in kids, the influenza B virus can cause abdominal symptoms such as a pain in stomach, vomiting, nausea, and some common problems which are not very resembles influenza, but it is rare and only for type B. After first stage of common symptoms, second stage will arrive, and it is a stage of respiratory symptoms which again lasts for 3 days. Again, any symptom of respiratory infection you will name is applied for influenza. Sore throat, running nose, some sneezing, coughing, dyspnea, tachypnea, it is fine for influenza as well. After stage of respiratory symptoms, convalescence will occur, and usually all symptoms will decrease significantly, and only some common malaise and some non-specific symptoms can persist up to even 4 weeks, especially in kids after common phase. So in general, even without treatment, spontaneous convalescence will occur, and people will become healthy in 7 days. And it is a common joke in medical community that if you will treat influenza, it will gone in 7 days. If you will not treat, it will go in a week. Mainly, treatment will not decrease duration of the disease. It can prevent mortality rate, it can prevent pneumonia, and severity of the disease can be milder. But with treatment, without treatment, common duration of influenza is 7 days. And disease itself, it's not so dangerous, but complications of influenza are important. And the major reason of death during influenza is pneumonia. It can be caused by virus, complicated by bacteria, but massive destruction of lung cells can lead to fatal pneumonia with high mortality rate. Rarely, disease can be complicated by heart damage or encephalitis, and again, mainly it is a pneumonia. One important thing, which is really important, not only in medical world, even for you, it is a complication known as a Reyes syndrome. It is described not only for influenza virus, but for some chickenpox and herpes viruses. and usually it is identified in kids up to 16 years of age. It was noticed that people who took salicylic acid for treatment of viral diseases have very serious systemic damage of multiple organs. And nowadays it is confirmed that this damage is related to destruction of mitochondria. And according to which system is affected more, symptomatics can be very different. We have destruction of brain tissues, of kidneys, of liver with corresponding failures. And actually mortality rate in rare symptoms can rise up to 40%, and usually due to some organ failure. So it's not possible to confirm and to prove something, because actually no one, of course, will check, especially on kids, if it's really so or not. But it was noticed that that syndrome is observed mainly in people who took aspirin or some other similar drugs. And then nowadays it is a common rule, and in some countries even it is an obligate law, that aspirin is prohibited in any viral infection in kids of first 16 years of age. And of course, it is a good measure, because actually mortality rate is high, and the only option to prevent disease is not to use salicylic acid. We don't have any specific treatment for that state. We don't have specific diagnostic tools, and it is only description of previous cases. And of course, no one again want to check, especially on you, if it really so or not. So again, common idea: if you have any respiratory disease and it's looking like viral disease, try not to use aspirin in kids. It's better to change to some other painkiller or antipyretic of another group of drug. If you talk about diagnosis again, and nowadays the main number of laboratories will perform PCR method as the main routine method to confirm the diagnosis. Of course, main number of people will not go to a doctor. They will be put it with some symptomatic diagnosis, and usually epidemic in the city or in some area, and symptomatic or symptoms of respiratory infection is enough to put the diagnosis. Of course, if you need to confirm by any reason, you need to be sure that it is influenza or it is important for some documents, then we can have immunofluorescence as the reference method. You need to take some respiratory sample from subject in the first 3 days of the disease while virus is present in the epithelium, and treat it with immunofluorescent antibodies. In a microscope, you will have that respiratory epithelium cells which are rich with viral inclusions. And rarely we can use complement fixation test or hemagglutination to confirm the diagnosis. So immunity after disease or after vaccination is humoral mediated and usually lasts not more than 2 weeks. 2 years. That's why a vaccination against disease can be done annually in perfect way. So again, we said already that prevention of disease is difficult mainly because of very high rate of antigenic variations, and actually we have a lot of different types of vaccines. We will talk later about formalin-killed vaccines which were used previous years and nowadays are not used because they have very low efficacy in respiratory immunity. And live vaccines which were used and still sometimes used nowadays are kind of dangerous because they can give a rise for disease. So influenza is one of the diseases, viral diseases, where we have some therapeutic options. And actually we have the classic drug, which is rimantadine and amantadine, which are very good in prevention and treatment of the diseases both. And again, in the contrast with antibiotics, antivirals can be used for prevention as well. We don't have any virus in composition of normal flora. It's not possible. They obligate parasites. And of course, if you take antivirals, they are not affecting, they cannot affect bacteria. So normal flora are not affected. And of course, structure of virus is absolutely different than structure of human cells. And those drugs have very low rate of side effects or adverse effects. So amantadine and rimantadine can be used for treatment as well, but nowadays very high rate of resistance is observed, and actually even if virus is initially sensitive to the drug, resistance can develop even during one episode of pandemic or epidemic rapidly. So one of the newest drugs is zanamivir. It is available in almost every country, and that drug can block virus viral enzymes, especially in neuraminidase. And of course, it is useful in prevention of the diseases as well. If the drug will accumulate in your tissues, in your blood, in your epithelial secretion, then virus cannot infect, cannot destroy cells because neuraminidase will be blocked before stage of infection. But till nowadays, especially during pandemic period, drug is pretty expensive and costs a lot, especially if you will need to give it for every family member. One specific test which can be and which will be asking you in exam, especially by some other elder teachers, it is an inhibition of hemagglutination. Test is absolutely primitive by idea, but you're always confused with it. I don't know by which reason. So if you will think, then influenza virus have hemagglutinate. It is an enzyme which can cause agglutination of blood cells. So idea is very simple. If you need to confirm that you have influenza virus, take that virus mixed with red blood cells. If virus is present, red blood cells will be clumped. And actually, in laboratory, the test should be performed in specific plastic plates, or we have commercial plastic test systems. It is a small microtiter well, which is filled in with the sample and reagents. So take swab from the subject containing virus, take diagnosticum which contain erythrocytes, and mix. If agglutination is observed, then it is virus which is present. It is common reaction of hemagglutination. And now you need to inhibit agglutination. Means that you need to take antibodies to influenza virus and mix with the clinical sample of the subject. If it is really influenza virus, antibodies will block enzyme, and when you will add red blood cells, agglutination is not occurred, and you will have formation of sediment in your microtiter well. So in the first picture, number 1, 2, and 3, it is agglutination of blood cells by virus. Means blood cells cannot form a sediment, they will be spread in the solution and they make some kind of a web. Blood cells are fixed by the virus and they all swimming in solution in equal manner. But number 4, 5, and 6, antibodies didn't match. Antibodies blocked the virus, and blood cells cannot be clumped, they cannot form agglutinate. And they will make a sediment, they will fall down on the bottom, and they will accumulate as a small bottom in that well. So actually, if you mixed virus or sample from subject with the diagnosticum containing red blood cells and agglutination was present, add antibodies, agglutination is absent, test is positive. Antibodies recognized virus, virus is influenza. So that test can be used in another modification when you need to identify antibodies in subject serum. And in that case, you need to evaluate immunity. Usually it is done twice during disease, and the beginning and at the end of the disease. Titer of antibodies should rise. And in that case, you need to take initial sample, mix with antibodies from the subject, mixed with known diagnosticum of virus containing viral particle and blood cells, and see if agglutination is present, dilute serum of the subject twice. And in first slide 1, 2, 3, 4, 5, 6, it is a serial dilution. Initially 1, then to 10, to 20, to 40, to 80, to 160. And in that case, you can see that initial titer is significant to perform agglutination, and number 2 agglutination is present, 3 present. In fourth well, titer of antibody is not significant to perform that agglutination. And actually, blood cells form a sediment. And by knowing which dilution is used, you will identify how many antibodies are present. And in a week, you will repeat the test. If titer became higher, means more number of antibodies appear. It is the beginning of the disease. Antibody are rising, and it is really that disease. If titer is stable, maybe previous disease or subject was immune. So in lower picture, you can see that in type C, in the row C, antibodies are not present at all, and in every well you have that formation of sediment or blood cells. In first two rows, in A and B, 1, 2, and 3 wells have hemagglutination. In fourth well, agglutination is stopped or inhibited. So the key idea to prevent influenza, it is vaccination. And actually we have huge different huge number of different types of vaccine. And actually any type of vaccine you can name, influenza vaccine exists. We said already we had formalin-killed vaccines, we had a live vaccine, we have recombinant, some units treated. Any type of vaccine is available. So two most popular vaccines are presented here. It is Vaxgrip and Influvac. Both are inactivated vaccines containing four types of virus. Usually it is H1N1, H3N2, and two B lines of influenza virus. It is Victoria and Yamagata. They are used intramuscularly, usually in the forearm, and according to manufacturer and according to different countries. They can be licensed for kids beginning from 6 months of the age. So in different countries, regulation can be different, but the youngest possible age for vaccination is 6 months. And as we said already, those vaccines should be used every year. Initially, the first vaccination should be done with the period in a month, means 2 doses initial and a booster dose. 1 dose today and a booster dose in a month. And after that, every year, 1 dose of the vaccine should be provided. Again, they will provide strong immunity only against viruses which are included in the vaccine. But of course, if it will be any strain which contain even one antigen of the same structure, it can be not H1N1, it can be some H1N5. Then subject will have partial immunity because H1 is the same. And the main task of the vaccine is not to prevent the disease itself, it is to decrease mortality. And actually, again, maybe rate of influenza is not decreased significantly, but number of lethal outcomes after vaccination decreased. Second type of vaccine which is used, and nowadays even in some countries, it is spray vaccines which contain a live virus. They can be monovalent or polyvalent, and they are administered intranasally. The main idea that vaccine is more suitable for kids. You don't need to perform injections. Kids are scared. Parents can refuse a vaccination because it is a needle, it is a pump, it is painful. But that one is an inhalational vaccine. It is delivered as a spray, and actually they are licensed for only healthy and non-pregnant individuals from 2 years and up to 49 years. Elderly people should not be vaccinated with that vaccine because it is alive, and if immunity is impaired, and usually during with the age it will be impaired. That vaccine, as we said, can cause disease even by vaccine strain. We have vaccine with adjuvants. It is available in some countries, and it is indicated for elder people 65 and above. That adjuvant will increase immunogenicity of the vaccine without increasing of viral load. Means that number of viral particles is less than a usual vaccine, but thanks to adjuvant, immunogenicity is equal or even higher than in other types of the vaccines. So usually that vaccine as well is quadrivalent, means contain 4 types of influenza virus. One newly vaccine, it is cell-based vaccine. All other vaccines are obtained with the help of virus cultivated on chick embryo. And the main idea of cell-based vaccine, that virus which is grown in mammalian cell culture have more number of similarities to wild strain. It means that pathogenicity is more equal to wild strain, and all properties of the virus are more close to native type of the virus, and that vaccine should have better preventive effect than common vaccines. But again, that vaccine is limited in use and approved only in few countries. In our country, that vaccine is not available. By manufacturer, it is stated that vaccine is indicated for kids from 4 years of age. It should be pretty safe. One recombinant vaccine, which is known as a flu block, I found out in that one vaccine. It is common vaccine of influenza, which is obtained by genetic engineering. The same quadrivalent vaccine, but it should be more safe than common vaccines which are using alive or complete viral particles, because they contain only antigens or virus without complete viral particles. Again, it is mainly in research and development stage, and till nowadays in some countries it is licensed for adults only. Actually, that's all about influenza infection, and very few words about parainfluenza virus. As you can see from the name, that virus is very similar by structure and properties to influenza virus. Actually, it belongs to the same family, have the same absolutely properties, and described in 5 types. Parainfluenza virus 1, 2, 3, and fourth one is split into A and B. All peculiarities of clinical symptoms of clinical presentations are again identical to common flu, but diseases are less severe. Usually they are very mild, self-limited, and can occur as infection of upper respiratory tract for first 2 types of the viruses, or lower respiratory tract for type 3 and 4. Again, people usually will not be diagnosed in proper manner, because common diagnosis of respiratory viral infections will be provided for those people, or doctor will just say that it is flu and put diagnosis of common flu. So disease is not so important. Little outcomes are extremely rare, and it is the reason why we don't have any specific therapy. We don't have vaccines, and usually people will have very mild disease in a few days, which will self-resolve without any management, without any treatment. And diagnosis will be done symptomatically, and of course, under diagnosis is the common thing for that virus, because of very low significance in human community.